I'm here to present on behalf of Rafael Perez, who couldn't came because he did not get the visa, but I'm very happy to be here with you. And I'm very happy that you set this challenge because it was very interesting for us. So yes, I'm going to present you about the Gut Microbiome Health Index Challenge. We all know that gut microbiomes play a crucial role in human health. Since the 1885, E. coli was isolated for the first time. There are a lot of diseases that we believe that are related with microbiota. We don't know if it's the cause or the consequences, as you know, but there is some relationship between microbiomes and human health. So CAMDA has a history in metagenomic classification challenges. For example, in 2017, we have 16S. In 2018, you presented whole genome sequences, and then we tried to classify the metagenomes using functional classification. And in between those years and 23, also it was included antimicrobial resistance. And the point is that we are trying to use these features. In that time, it was for classifying microbiomes from the city of origin. And now this year, we are attempting a slightly different classification problem. We are trying to classify microbiomes according to their relationship with disease or health. Of course, this is different because disease is a lot of diseases. It could be different bacteria that are related to some specific disease. And we are hoping that there is something that really captures what it is a health people, even if they are from different environments. Okay, as we have just heard, microbiome health index are different approaches to microbiome health indexes. The first one relied on taxonomic profiles. For example, just alpha diversity. If you have many different species and a different distribution of them, or perhaps the gut microbiome health index. This one, it's about how many good or bad microbes do you have. It also in 2023, we have the high dimensional PCA, and this one it tries to measure the contribution of each bacteria, but all of them are just about the taxonomical content of the microbiome. We also have some web tools to visualize the gut microbiome sample, your gut microbiome sample against, for example, 4,000 samples are available. This is a work from 2003. Here is the sample, and it tries to localize and visualize the sample against other gut microbiome samples related with health and disease. But we want to move far away. We want to take also into account the functions. And this is what we found two indexes that are trying to consider it not just the taxonomical content, but also the functional content. This one is the one that we just heard about, Q to dysbiosis. And the other one I found is the alpha dissimilarity, diversity, and this one is trying to account not just the functions that are present in the microbiome, but also how divergent are each gene family in that microbiome. So if it is very divergent, many families, well, perhaps they can a broader range of functions. So the data evolve, and also our way to approach to the data and the index are evolving. And so this year CAMDA challenge were to develop a superior gut microbiome health index using all the theater concept of the functions and the relationship between two bacteria, and also to use the developed index to identify a cohort of COVID-19 patients differentiated between healthy and controls. Okay, so we received 630 samples from the American Gut Project and the Human Microbiome Project. We, from these samples, 300 were healthy, 300 were unhealthy. And we also got pre-calculated indexes, the Shannon entropy, the gut microbiome health index, and the high PCA. The unhealthy were divided in obese, Crohn's disease, and ulcerative colitis. And we also got the taxonomic profiles and the functional profiles. So we have the data, the features, and we also know the classification labels. Just as a first exploration with taxonomical indexes, we got the alpha diversity, which shows that Crohn's disease and ulcerative colitis have lower alpha diversity. But the same problem, the obese samples were not separated by beta diversity. You can see the obese are the green one and are distributed along all the samples. We cannot separate those samples. So we decided to start by adapting existing taxonomy classification methods for functional profiles. So we started with the gut microbiome health index. This index is about to choose some bacteria that are more related with healthy people and to choose another subset of bacteria that are related with unhealthy people. And what you do is you just calculate some proportion and differential abundance of these bacteria. If the ratio of the bacteria of one species that it is present in more healthy people and unhealthy people is greater than some theta F value, and also the difference between them is greater than some value, then you classify this bacteria as a good bacteria. And if it's different, if it's the other way, then if it's greater, the unhealthy, the people who is unhealthy and contain that bacteria and the difference, then you classify that bacteria as unhealthy related bacteria. Then you calculate that proportion, the healthy related organisms and the unhealthy related bacteria. And if that index is greater than zero, then you are healthy and otherwise you are unhealthy. So you calculate a distribution, and with that idea, you decide if that sample was healthy or unhealthy. Okay, well, what we did is we do the same, but now instead of using only bacteria, we use functions. So we try to associate functions with healthy and unhealthy samples. Because of that, we use the Metaflan taxonomy, the human the human pipeline to get the functional annotations. And with integrated and unintegrated pathways, with these three features, we calculated the gut microbiome health index, and we run a random forest classifier to try to predict these two levels, healthy and unhealthy. By the way, the gut microbiome health index was in R. We tried to homogenize everything in Python. I know that you gave all the index, but we recalculated in order that we can do some modification because perhaps that way we could improve a little bit the indexes. So we moved everything to Python. So we recode everything. So in that way we can benchmark all the indexes, and we are trying to having perhaps some library, and if we are lucky, some visualization like the one that I presented before at the beginning, when you can compare your sample against all the index, but not just the taxonomical index, but also with the functional index. That would be our idea. Okay, so with these improvements or slightly modification of the gut microbiome index, here is what the accuracy that we got with the given gut microbiome health index. And after the code readaptation to Python, we got 76 because we also adjusted some parameters. And but these are just with the taxonomy, the first two. But when we add all the pathways, the integrated and unintegrated pathways using the human, we are after combining the two characteristics, not just the taxonomical, but also the functional features. We got an 89.3 of accuracy. I think we can be a little over adjusting the data. But anyway, in the past CAMDA challenges, when you combine the taxonomical and the functional features is when you get a slightly better classification than if only you use the taxonomical features. For example, with taxonomical classification, you can only decide between cities until an 85%, for example. And if you add these new features, considering the functional features, you always get a little better. Functional features perform lower than taxonomical features if you only consider them separately, but together you always get a little improvement. Okay, so we have finished with with this index. So now I'm going to talk to you about high PCA. High PCA identifies key features and generates statistical distributions based on principal components. So first, you start with the functional annotation with human pathways. You have two, the taxonomical from Metaflam and the human for the functional annotations. And then there is a step of feature selection where you can use Kolmogorov-Smirnov or you can use differential abundance. After that, you do a data transformation to some standardization and scaling. And then you do the principal component analysis. Well, after you calculate some statistics using Hotelling or Q statistic, and this is a hypothesis testing. If you are in some side of the distribution, you are healthy, considered healthy or unhealthy. So again, what we did is we code this in Python. I don't know how you run it because it was in MATLAB at the beginning, and we tried to translate because it was difficult to run it. It didn't have functions, so we did this. Well, not me, but Rafael did all the work to have something that it was more reproducible, so all the indices can be compared now. And okay, we variated the way that we choose the features. So for example, this is Kolmogorov-Smirnov, but with 18 or 130, healthy and unhealthy. We also changed the distribution value, if you are going to use only 0.05 or 0.005. And this one is also considering the pathways. This is just a taxonomy and considering the pathways. And this is all we got, 80%, 80.9, the best that we could get. Well, this is before combining taxonomy and functional. But then we could, what we did is we glue these tables, taxonomy and human features, run some standardization, and after a logistic regression. This is what we have until now. All suggested methods achieve better when you combine the functional and taxonomical features. This is the one that you gave with the R, 62% of balanced accuracy. Then when we run it in Python, we get 8.9, and the high PCA, the one that you provide gave us 70%. And with the Python modification, the Kolmogorov-Smirnov feature selection, and the logistic regression to combine functional and taxonomical characteristics, well, we get 82%. So this is what we got in terms of the healthy index. So now I'm going to talk to you about the next challenge. We didn't know that people could get better in the COVID, so we consider that you were healthy, you were sick, and then you always get healthy. So this is a surprise to me, but okay. We evaluated the proposed methods in the COVID-19 gut samples. We got 35 patients. We didn't know if they were sick, the red ones, or healthy. And we got 2 samples. One is the first day, and the other one is the last day. So the first is just a beta diversity visualization. So here you have the samples. The circle is the first day, and the triangle is the second day. Well, we believe that the ones that change the distance the most are going to be the ones that are healthy and then, well, sick and then get better, because they are supposed to be changing the microbiome if all that we are talking has some sense. Because, well, microbiome should change if you change your healthy state. Okay, so we believe that these ones are the ones that are changing from healthy to unhealthy, or the other side, from unhealthy to get worse, perhaps. But anyway, we calculated all the indexes for each sample. For example, the high PCA with the Kolmogorov-Smirnov, the GHMI, the beta distance, and we have our prediction, healthy, unhealthy. Many of the samples have a consensus if they were healthy or unhealthy. But this is what we believe. We are not sure if it is true or not. We believe these ones are the control and these ones are the COVID-19. But we have 2 cases where we were not sure, because this one first were healthy and then was unhealthy. So, and there is a change over there, and it was impossible to us because we believe that you always get better. So now that you say that, perhaps this is one of the cases where you were sick and then you get worse. I don't know, perhaps there are more, but I believe that this could be one. And in this other case, one index says that it is healthy and the other one says that it is unhealthy, but there is not a lot of change in the beta distance. So the 2 microbiomes are really similar. So we classified it as healthy because we believe that you couldn't get worse again, but I don't know now. Okay, this is what we did in terms of healthy indexes. But also we try some extra analysis, because this is the spirit of the challenge, that you can get creative and think about something different, whatever information you can get from the data. So we try to reprocess again all the pre-processing, because perhaps having some better classifications of species, because having just Kraken, for example, it could give you a lot of noise if it is a species or if it is a genera. So we decided to run Bracken, to run to taxonomic assignment, and we also run several set of annotations. Here we have PROCA, but we also run another functional annotations like MetaPsych and other ones. And for this, we tested this index that I told you before, the one that not only considers different functional annotations, but also how divergent are these families of functional annotations. Because it says that if you have similar species, well, perhaps you have a lot of functions, but if you have that same functions, but with the species that are different, divergent in the tree of life, then the functions that you are having perhaps are broader, because it's going to be divergence in these trees. Okay, this one adds to alpha diversity, not just the content, but the dissimilarity of each gene family. And with this index, the obese feature, well, metadata, it's higher. I don't know if it's really, if we can really differentiate, but it is consistently higher than the other groups. Finally, where we were trying to choose our own important features and our own important bacteria, not just using the association, but perhaps looking for key species or for key functions in the sense that they are more related or connected with other functions in healthy or unhealthy microbiomes. And well, we discovered two highly dense subnetworks associated to Crohn disease. These networks, this is the bacteria that contains. Well, I have been hearing these names during these 2 days, Laconostridium, Ruminococcus. I haven't heard of Laconoclostridium before, so I guess, well, perhaps it's something relevant. This is the really connected species. We need to investigate more about it. And another set of ideas, something else that we did is to look for the topological data structure of the data. A topological data analysis tries to infer a more general structure. For example, here you have a hand, but perhaps you don't need to look at the whole hand. You only need to know the structure. And the structure is like you have these 3 fingers and, well, this bigger form. If you want to know more about TDA, Jaime is going to present his poster at the 118. And we have a carpentries incubator lesson. We really love to do some teaching. And we believe that the carpentries is a good standardized way to produce lessons. We have a lesson to start metagenomics. And we would love to include all the statistical procedures that we need for CAMDA. Okay, if you want to know more about TDA, please visit Jaime's poster. We use this topological analysis. In particular, we use TMAP. TMAP, what is a reduction of the number of points. Here you have, for example, a lot of microbiomes in that square, but perhaps you can take only 1 point in the blue class, 1 point in the green class, and represent all the other squares with 2 red dots. This is a circle, and you only need that to describe the data. Then SAFE is another algorithm to correlate the network structure to a variable. Here this is a reduction of the microbiomes, and we colored according to the high PCA index. And the SAFE is looks the correlation of the variable with the network. If you get a lot of blue, then there is really not a strong correlation, but in the places where you are more red, for example, here, well, this is a subnetwork that is red, and it is almost full of Crohn disease microbiomes. So the take-home messages is that we adapted existing indexes to work with functional profiles, integrating taxonomic and functional profiles. And that now everything is in Python and we can benchmark these indexes. We tried to do it with Q2 dysbiosis, but we couldn't run it because we missed one file, the human unstratified file, and it was needed for run the index. So once what we have it, we will compare it. Okay, our proposed methods, our proposed modifications of the methods slightly outperform the provided metrics. Using the proposed methods, we attempt to distinguish controls from COVID-19 patients. And we also found this index that considers the divergence in the families and the topological data analysis, and we will try to include this in our research that is going to be last the next year. So please, if you have any suggestions or if you have something that you want to tell us, please do it now. All the code is in GitHub, and we created a Docker container so you can reproduce all the flow. Everything is in Python. Rafa, I thank Rafa because he really wanted to be here, but okay, I thank all the team who work with us. This is the team. We are a team of mathematicians and biologists. And this is a class that we created, and we found that CAMDA challenge was really perfect to join people from mathematical, computational, and biological backgrounds. And we took a statistical class all the semester, and at the end, we finished this as the project of the class. So these are the three teams, because we participated in the 3 CAMDA challenges this year. So I thank you, all of you, and that's all.
Thank you, Nelly, for this very and really enriched analysis. We might have time for 1-2 questions. I guess definitely we would like to chat with you more during the break about your analysis results, and probably would like to also. Yep, you want. Okay, now just chat about all these things which you rewrote because they are not reproducible, unfortunately, the PCA, right? So yeah, it will be very interesting to discuss and work together about using this stuff further. Okay, so yes, you wanted to ask a question.
Hello, very nice talk. I just have a short question. You mentioned that when you analyzed this, you were not aware how the health status of the patient could change. So now that you know, how does that affect your interpretation, or how would you change your analysis, if at all?
Well, the first change is that now I think that the cases that we are sure that we were wrong, perhaps we were not wrong. That was a better explanation, that you were in a healthier state and then you become a little unhealthier. Yeah. And well, I could try to classificate at a classification level, right? Because just before I was just using 2 labels, healthy and unhealthy, and now I think I would use 3, healthy, unhealthy, and worse. Yeah.
Okay, so you will have a nice update for the proceedings, right?
Uh-huh.
Thank you.
Thank you.